everybody, it's Miss Chatzy again. How are you? Hope you're doing well. Wish I could see you, but I can't. Welcome to the very final topic, brand new topic of the entire year. So our last topic of the year piggybacks off of what we've done in photosynthesis. So photosynthesis was all about how autotrophs can make glucose molecules using the sun as the source of energy. So our last chapter here talks about what happens to the glucose molecules once they are made. So let's say I'm a little plant, I've made this glucose molecule, what am I going to do with it? Well, usually people equate photosynthesis with plant growth because that's a really good way you can measure whether or not it's happening. So sometimes glucose molecules are combined together and rearranged to form other larger carbohydrates, things like cellulose, for example, which is what cell walls are made out of. So if a plant wants to grow, it's got to do mitosis and it's got to get the raw materials from somewhere. So those raw materials would come from combining of glucose molecules. We call that biosynthesis, like when you make new biological molecules. So that's one of many options, but there is another thing to consider. Sometimes, because that glucose molecule has a lot of potential, chemical energy stored up in it, sometimes the cell is in need of energy for other things, like maybe it's doing something besides just growing. So cellular respiration talks about how the cell can take a glucose molecule, break it back down into smaller pieces, and extract energy from it. So in the diagram on this page, what you see is your little glucose molecule entering a cell. This big blue object here is our cell. And then through a couple of step processes, we're going to be using the mitochondria to produce a whole bunch of ATP. So if the cell has to do something that requires a large, input of energy, this is where it comes from. So long ago, you learned that the mitochondria is the powerhouse of the cell, which is kind of a stupid definition to teach kids because it doesn't really explain what a powerhouse is, but it's basically the location in the cell where all of the energy is going to be released. So if a cell needs to do any kind of work, this is where that energy is going to come from. So basically, cellular respiration describes how cells, and by the way, this includes both plants, animals, fungus, protists, anything that's a eukaryote, how it is that we break down our food and actually get energy from it. So just keep in mind, really importantly, that plants do this as well. So plants do both photosynthesis and respiration, whereas animals like us only do respiration. We have to get our food from other sources because we are heterotrophs. Okay, so I'm going to walk you through the steps of how it is that it actually generates the energy. But I first want to talk a little bit about the similarities between the chloroplast and the mitochondrion. So mitochondrion, by the way, is the singular of mitochondria. So it's usually a little bit of a kidney bean shape, and very much like the chloroplast, it too is a double membrane. And when we talked about as being the benefit of being a double membrane with a chloroplast also applies here. If you've got multiple layers inside, you have extra surface area. And the more surface area you have, the more chemical reactions you can do upon that surface area, which is basically an efficiency issue. Like the more membrane you have, the more efficient you are at moving things across a membrane in a short amount of time. So that's going to play a role in the same way it did in the chloroplast. And it's all about making ATP. Structurally, just a couple of things that I don't know if they're really significant, but just in case they're ever labeled on a diagram. The little folds that you see, these are what's called the cristae, so that's where the gradient is formed and things cross that little membrane. The matrix is the fluid that fills up the inside of the mitochondria. And then also, interestingly enough, not really super related to what we have been doing, but notice a new type of DNA that you may have heard of or may not have. It's called mtDNA, mitochondrial DNA. Just like the chloroplast, the mitochondria has its own set of DNA. So when mitochondria replicate, when you make more of them, they do it on their own. So it's separate from your regular nuclear DNA. Mitochondria have their own genes to replicate, and then all the proteins that are necessary to make the mitochondria operational are produced here. there's about 13 proteins that are necessary to make respiration happens. And also, fun fact, your mitochondrial DNA most likely came from your mother, and it has to do with when the sperm and the egg combine during fertilization. I believe the sperm mitochondrial DNA disintegrates, for lack of a better word, and the mitochondrial DNA becomes your own mitochondrial DNA as well. I think I said that right. I hope that's true. Let's pretend it is. Okay, so like in photosynthesis, we had a 2-step process. In respiration, we're going to have a 3-step process. It's not really more complicated because a lot of it is very similar to what we saw in how the chloroplast makes ATP. So in eukaryotes, meaning anything that has a nucleus, including plants, we're going to make ATP by breaking down carbohydrates. So for you, when you eat food, you're probably not eating glucose, my guess, but you're eating other things that are going to be digested, broken down, and rearranged into really simple carbohydrate molecules. So ultimately, whatever you eat, like if you ate a big bowl of pasta, you're going to break a lot of that down, if not all of it, into glucose at some point. So the glucose is in the bloodstream, and then that's going to diffuse into the cell via a glucose transporter on the surface of the cell membrane. That's going to transport the glucose molecule across because it's too big for regular old diffusion. Once it's in there, there's going to be a bunch of reactions that take place that are driven by different kinds of enzymes. I don't think you have to know any one specific one besides the ATP synthase, but remember that enzymes always end in ASE, and the job of an enzyme is to speed up the chemical reaction. We're going to talk primarily about what's called aerobic respiration. The word aerobic just means oxygen. So if you've ever wondered why it is that you need oxygen every day of your life, all day, in order to survive, this is why. So we all kind of recognize that oxygen is necessary, and without it, your cells begin to die, and then your body begins to die over time. So the reason why oxygen is needed is because it is used in a very important step in aerobic respiration inside the mitochondria. And if oxygen is absent, then the body and the cell in particular, cannot make ATP, and then it runs out of ATP and then it dies. So hence the reason why oxygen is necessary. So we have a 3-step process here. We're going to talk about step 1. I think is the most unique one. You're more than likely to get a question on the AP exam, if you were, about glycolysis, because it's the one that's kind of universal, that even bacteria have a hand in glycolysis. Then we have something called the Krebs cycle, named after Hans Krebs, and then electron transport, which is something we just talked about in photosynthesis. It's truly the same process. Okay, here's a little overview, including the equation. If you've got a little sense to you, I'm hoping that you notice that the equation is simply just the reverse of photosynthesis. So what you see here as being the reactants, those were the products in photosynthesis. And then what you see on the other side as being the products in respiration, those were the reactants of photosynthesis. So we truly just kind of flip-flop our products and reactants there. And of course, I know that ATP is not actually a molecular chemical. It is energy, which is not quite the same thing, but for the sake of memorization or just kind of associating things, we'll leave it as one of our products. So in the picture at the bottom there, you've got the whole mess of kind of semi-organized. Notice there's a couple of important things. Part of respiration happens in the cytoplasm. That's the glycolysis part. And the other part of respiration, actually the other two parts, happen in this little yellow bean here, which is supposed to be the mitochondria. So some of it takes place in the cytoplasm and some of it takes place inside the mitochondria. Okay, so let's talk a little bit about step 1, glycolysis. First, just to break down the word itself, the suffix lysis, or to lyse, means to break something down, to break the bonds of it. Glyco, if you remember from we were talking about diabetes, and we were talking about muscles, sugar storage in muscles, glycogen, it's just a word that refers to sugar. So glycolysis means to break down sugar. So in the very first step, the sugar molecule diffuses or transports across the cell membrane into the cytoplasm, and we are going to break it apart, pretty much. We're going to do that by rearranging all of its bonds. So we're not going to break it apart and blow it away. We're going to break it apart and rearrange it into a new molecule, a temporary molecule called pyruvate. Sometimes it's called pyruvic acid, but you do not need to know the details of what pyruvate is. You just have to know it exists. When all is said and done, glycolysis is going to give us a net gain of 2 ATP molecules. It's not a lot. It's definitely not enough for you as a large animal to survive on, hence the reason there's more steps. But some organisms can survive for temporary amounts of time just with a small amount of ATP. FYI, we actually produce 4 ATP, but it took 2 of them to break the glucose bonds. So to make pyruvate, you have to actually invest some of that ATP ahead of time. So here's a little diagram showing you what we mean by investing ATP. So when our glucose first is in the cell, 2 ATP are going to go in, and that is going to be used to break apart the glucose molecule and rearrange it. The breaking of those bonds is going to cause the release of 4 ATP molecules. We're also going to produce 2 electron carriers called NADH. Fun fact, it is the exact same molecule as NADPH from photosynthesis. The only difference is the P stands for photosynthesis, and since we're not in photosynthesis, it's just called NADH, but it is the exact same molecule with the exact same purpose. In the electron transport chain, it's going to steal some of those electrons to remove them from the chain. And then notice we also make 2 molecules of pyruvate. That pyruvate is then going to be transported into the mitochondria, where we're going to break it down and rearrange it, oxidize it, move all of its electrons around, and steal as much energy as we can. So I mentioned before how glycolysis is probably, well, it used to be a really significant point of the AP exam because there's a really good tie-in to evolution. What we're pretty sure about is that the first prokaryotes on Earth, meaning the first bacteria, used some form of glycolysis to make their energy. Because even though bacteria don't have mitochondria, they still need energy to survive because they still do, a lot of normal cell behavior, like, binary fission, like copying themselves. How do they make more bacteria? Well, they need ATP to do it. So we're pretty sure that glycolysis is one of those chemical reactions that's common amongst all living things, even if you don't have mitochondria. Some of the evidence that supports this idea is that glycolysis does not require oxygen. If you noticed, it wasn't mentioned in any of the previous slides in glycolysis. And that's because it has nothing to do with the mitochondria at all. And also bacteria, which probably exist, let's say 3.5 billion years ago, we're pretty sure there wasn't even oxygen on Earth at that time. So it would make sense that these little prokaryotes evolved on a planet without oxygen. So the process by which they make energy must have also been present in the absence of oxygen. That was hard to spit out there. So we're pretty sure that because there was no oxygen 3.5 billion years ago, that glycolysis is probably the common denominator that we share with those old and current bacteria. Also, it's a very widespread, meaning that everybody does it. Everybody does it, even since the very early organisms on Earth. We assume this is how they got their energy. And the fact that glycolysis happens in the cytoplasm. Cytosol and cytoplasm are not exactly the same word, but they often refer to the same location. The cytosol is the fluid, the cytoplasm is like the fluid, and then when you put all the stuff in the fluid. So in other words, glycolysis doesn't require organelles, and that ties back into our evolutionary idea because the first cells, the prokaryotes, did not have membrane-bound organelles. So how would they have gotten energy? Well, they could still do glycolysis because glycolysis only requires cytoplasm.